Hello, my name is Kathryn Atherton, and I am a student in the Bioinformatics graduate program at Boston University. Today, I'm going to be talking about my group's work entitled APOEE4 is Associated with Chronic Traumatic Encephalopathy, or CTE. CTE is a neurodegenerative disease defined by deposition of perivascular hypophosphorylated tau, or p-tau, in the sulci of the brain. You can see the stained perivascular p-tau lesions in multiple brain slices in Figure 1. Symptoms of CTE include memory loss, problems with problem solving, explosivity, depression, and difficulty with motor skills. The major risk factor for CTE is repetitive head impacts, or RHI. However, people with the same RHI exposure develop varying severities of CTE, and some don't even develop CTE at all. This suggests that other factors, including genetics, might play a role in this disease. Alzheimer's disease is also defined by the aggregation of tau in the brain, but in a different anatomical pattern than CTE, as well as amyloid plaques. The APOEE4 allele is a risk factor for Alzheimer's disease and is also associated with poor outcomes after traumatic brain injury, or TBI. Because of these relationships, we tested whether APOEE4 is associated with CTE as well. To do so, we used the largest dataset of pathologically confirmed CTE cases with matching controls. It is important to note that because this is human data, collecting controls that match the cases in terms of contact sports played and duration of play is difficult. We have 535 subjects, 295 of which were adjudicated to have CTE, and all of whom had substantial exposure to RHI, mostly from contact sports and military service. All of these subjects were genotyped for the major APOE alleles, and we calculated their dosage of the E4 allele. Additionally, we measured the tau burden in their brains, both quantitatively, based on digital slide scanning, and semi-quantitatively, based on neuropathologist assessment across multiple brain regions commonly affected in CTE. For the quantitative tau measurements, a computer algorithm analyzed digital images of brain slices stained for p-tau and calculated the density of p-tau in various brain regions. For the semi-quantitative measurement of tau burden, a neuropathologist assigned a level of severity from 0 to 3, 0 being no tau burden and 3 being the most severe tau burden in various brain regions. We used logistic and linear regressions to analyze the relationship between APOEE4 dosage and CTE and its related phenotypes. All of these regression models adjusted for the subject's age of death and their self-identified race. Additional models also adjusted for neuritic and diffuse amyloid plaques to assess the APOE relationship independent of Alzheimer's-related pathology. CTE-related phenotypes included diagnosis of CTE, which means does the subject have CTE, yes or no? CTE stage, a measure of CTE severity from 0 to 4, and the quantitative and semi-quantitative measurements of tau burden in various individual brain regions, as well as in the entire brain. The results of the models predicting outcomes from the entire brain that only adjusted for APOEE4 dosage, age, and race are shown in Figure 2. As you can see, the model predicting CTE diagnosis was not significant, but the models predicting CTE severity via CTE stage, the total sum of quantitative tau measurements, and the total sum of semi-quantitative tau measurements did show a significant relationship with APOEE4. We believe the reason that there was not a significant association with CTE diagnosis is because analysis of this binary trait has less statistical power compared to analyses of the quantitative and semi-quantitative traits. Furthermore, when adjusting these models for amyloid plaques, as shown in Figure 3, we see small decreases in effect size, but the effect is not entirely removed from most models. This means that the effect of APOE on CTE-related outcomes is largely independent of Alzheimer's disease and amyloid-related pathology. Figure 4 shows the results of the models that predicted tau burden in the individual brain regions after adjusting for APOEE4 dosage, age, and race. Figure 4a shows the results from models of regions measured quantitatively, and Figure 4b shows the results from models of regions measured semi-quantitatively. The top two most significant regions in 4a, the middle frontal crest and the middle frontal sulcus, are found in the frontal lobe, as are two of the top three most significant regions in 4b, the orbital frontal gyrus and the middle frontal gyrus. This tells us that APOEE4 dosage is most significantly associated with tau burden in the frontal lobe, which is one of the first regions affected by CTE. Other regions where tau burden is significantly associated with APOE are the inferior parietal lobule and the superior temporal gyrus, which are both cortical regions, and the entorhinal cortex, amygdala, and the CA2, 3, and 4 regions of the hippocampus, which are limbic structures. In conclusion, this is the most concrete evidence to date that APOE is associated with CTE. We have found that APOEE4 is significantly associated with CTE, even after adjusting for Alzheimer's-related pathology. Additionally, we found that tau burden in several different brain regions shows significant associations with APOEE4. Thank you for your time. Please come to my poster chat session if you have any questions.